purpose of today's webinar is to introduce some of the new developments that we've introduced into Pipsqueak and talk a little bit about how to use some of our features and how to get going with analysis in Pipsqueak. So what I'm going to cover today are, just some of our kind of high-level analysis capabilities like single label analysis, co-localization. I'll talk about how to do some more powerful types of analysis like batch processing of images. And then probably most importantly, I'm going to spend a little time talking about how to train a custom AI model in Pipsqueak so that its detection capabilities are really fine-tuned for your images. So again, feel free to ask questions anytime. Otherwise, I'll stop at the end for some Q&A before we hang up. So while we're here, just a little bit of background on Pipsqueak. As I mentioned earlier, Christy and I have been working with Pipsqueak since really 2016. We actually first published some of our methods when I was a postdoc at Washington State University. And since then, this program has really grown and evolved to be something much more powerful. So the incorporation of artificial intelligence to better detect some of the cells or objects that we're analyzing, is a huge improvement for its capabilities. And over this time, we've really seen a worldwide adoption of Pipsqueak, from users across the globe. So, it's pretty cool to see all the different types of analyses that people are doing with Pipsqueak. And it, I think is a good indication of its breadth of capabilities. So if there's something that you would like to do in Pipsqueak that you're not clear how to perform, please feel free to reach out to us because, we certainly have a lot of different use cases, a lot of different types of images or imaging modalities that are coming into Pipsqueak. And so, I'd be happy to see if we can point you in the right direction, if that would be useful. If you'd like to see some of the publications of people who are using Pipsqueak, you can find some of that information on our website. You can also find some citation information on our website. And I'll just point out here, we now have an RRID for Pipsqueak. So if you end up publishing with it, this would be the appropriate thing to cite in your publication. So nice to have that. If you're getting going with Pipsqueak, you can really find everything that you need to self-serve and get started on our website. So rewireneuro.com. From our homepage, just navigate to one of these Pipsqueak Pro options. A good place to get started is with our month-long free trial. So just navigate here. You can add this to your cart. You don't need a credit card or anything like that to get going. It will just ask you, your name and email sort of information and help you download and get started with it. We are in the middle of making some really exciting changes to Rewire. We're going to announce a little bit of rebranding on Friday. And so we're pretty excited about it. But if anyone has been trying to download Pipsqueak and noticed any issues on the website, I sincerely apologize. We're making a lot of changes behind the scenes. And so every now and then a couple things have gotten glitchy on the website. But this should be all set for you to download and get started with it now. If you have any trouble, just let me know. I'd be happy to help you get going with it. But we are pretty excited about it. Keep tuned or stay tuned and keep an ear out on Friday. We'll be announcing some fun changes. So with that, one of the things I do want to mention is a really cool collaboration that we initiated a few months back here with eLabNext, which is a part of the Eppendorf group. So eLabNext is Eppendorf's electronic lab notebook. And one of the things that we made possible was for users of eLabNext to automatically import their images into Pipsqueak and vice versa, export their data back into eLabNext. So it's a really great utility to store some of your lab material like protocols or data sets, share it and collaborate in your lab or even outside your lab. And so having this integration with Pipsqueak, I think, is really powerful because it makes it possible to seamlessly move your data back and forth. So this is a free integration. If you're already an eLabNext user, then you should be able to log right in from Pipsqueak and start making this integration possible. Again, if you have any questions about it, feel free to reach out to us. Actually, Christy, maybe you can pop our email into the chat right now. So yeah, feel free to ask any questions about that. So yeah, what we want to spend a little bit of time today is just walking through some of these high-level features and kind of getting going with Pipsqueak. So I'm going to switch over to my Pipsqueak interface here. If you are installing Pipsqueak for the very first time, it's gonna look a little bit like this when you get going with it. I already have an image opened, but this is generally our interface where you're going to be able to do everything that you need to do in Pipsqueak. On the left side, I've got a panel of my projects and some information about my projects. On the right side, I have all the tools and utilities that I need to use in order to perform my analysis. And in the middle, we've got a stage where we display our images or any type of image data information that we're looking at. Up on my toolbar, I'm on a Mac here, so this looks a little bit unique for me, but it shouldn't be all that different for anyone else. I've got some of my tools like logging in and out of eLabNext. I've also got some help keys here. So I'll talk about this in a second, and some tutorials, which are really useful if you're just getting going. This is also where I've got my ability to log in and out of Pipsqueak, to see my account, and to change some of my settings. And so one setting I'm going to point out here real fast, because this is going to look different if you first install Pipsqueak, is I'm using our dark mode. So this is pretty cool to be able to switch back and forth. Light mode definitely shows some contrast, but I think dark mode looks really cool. And it also makes it a little bit easier for me to see some of my cells and my images. So check out that setting as you're getting going. But great, so let's go ahead and get started with our very first analysis. When you start using Pipsqueak, we've got a project that's automatically imported here. That's kind of nice to start with. So this Pipsqueak Pro demo set is going to be imported, and it's going to have some images here that are guaranteed to analyze well. So it should give you an idea of how to use some of our cell detection capabilities and quantification, and then allow you to maybe start learning from there, or import your own images from there. So if I were to start with this project, I can do a couple different things. I could do a single label analysis here. And if I'm going to do a single label analysis, I'm going to choose one of the images over here on the left side. So here I've got what's called a WFA image here. This is actually some of the work that I did as a postdoc. So if this is my very first time in Pipsqueak, it's fantastic looking at this, but I don't really know how to start detecting my cells. Over here on the right side, I have a whole library of different AI detection models that I could apply to this image, but I don't really know which one is going to be the best choice. And so we developed a brand new feature called Recommend a Model. And this is pretty cool, because what it's going to do is it's going to take a look at that image, and it's going to compare it against a lot of the different AI models that we've already developed. And so it's going to start making some guesses about what is going to be the best choice for this. So you can see it's pulling up that same image and showing me a number of different AI models that might work for it. And it's kind of sorting the results down here as we go, and great, it's finished. And so what we've seen here is two of its top choices are this parvalbumin detection model and this WFA detection model. And that's pretty exciting, because this is a WFA image. So if this was my first time using it, I'd probably select one of these two as the model that I would use for it. The other thing that I like to point out from this is just a little bit of an example of how different AI models are detecting objects in this image. So as I scroll down here, I've got a model that's trained for DAPI. I've got a model that's trained for somatic staining. I've got a model that's trained for CFOS and microglia. You can see like these models really aren't picking up anything at all in this WFA image. And so, that's telling me that the features that these models are looking for really aren't present in the WFA image, or the features that it's finding are different than I'm actually looking for. And so that might help, kind of clue you in on why a model might be performing well on your image or not performing well on your image. So cool. I'm going to choose this WFA model. I can click into it. I can say select model. That's all great. So now I can make a mental note that if I'm analyzing this image, I'm probably going to use the WFA model. And I can find that here in my library just by scrolling down and clicking WFA. So this is good. It's pretty, it's detecting a lot of things, but it's actually detecting a little bit more than I might want to detect in this image. And so I've got a couple additional settings here that I can change to try to make my detection maybe a little bit more accurate for myself. So one of my detection settings is the sensitivity. And so this is about as it sounds. If I move my sensitivity down, it's going to detect fewer cells in my image. And actually, I may have gone too far. We might not detect anything at all here. So yeah, that detected nothing. I'm gonna bring it back up. I kind of like that 75-ish range. I've got another setting here for my overlap removal. And so as I increase this, what I'm going to see is there are fewer regions of interest that are overlapping across my cells. I do have a couple regions of interest that are overlapping, and I think that's actually probably appropriate in this case. I know just based on this cell type that there are a lot of gap junctions formed between these interneurons. And so, this is actually probably a gap junction that's formed here. I think this might actually be two neurons that are pushed up against each other. So, the overlap that I'm seeing here is actually probably accurate in this case. And so at this point, I think these detections are pretty close to what I might actually want to quantify. I have a couple more steps that I can take, though, to continue to improve the detection accuracy here before quantification. And that is, I can go in and I can draw ROIs just by clicking on the image and pressing down as I draw, or I can delete those. I can go into ROIs and I can change the size of the ROIs. I can also open up my toolbar here and change the type of object that's being drawn. So I might want to draw a rectangle, or I can use my polygon tool to draw something that's more freehand if that's more appropriate for my objects. So that's pretty cool. I can also toggle my ROIs on and off. And so if anyone's done this sort of thing in ImageJ, I'm sure you've used this functionality before. I find it really important to toggle just in order for my eye to kind of glance around an image and determine if I'm seeing what I want to see. The other hotkey that we have available for this, and again, your hotkeys are up here in your help menu, is Control-T. And that's going to make it really easy just to make those ROIs visible and then invisible. So, really cool to be able to do that. At this point, I'm going to say I like all these detections. I think it's pretty close to what I want. One more thing that I like to do before finishing my quantification is to subtract out the background of my image. And this is, probably a little bit based on, or going to be based on, the type of analysis that you do in your lab, and how you want to subtract background, is up to you. But in this case, I like to subtract background by using an automatic background quantification that happens in Pipsqueak. So I got to this by selecting my background subtraction and pressing set ROI, or set background ROIs. And here I can see these square ROIs around the perimeter of my image. And what these ROIs are doing are actually measuring the pixel intensities all around the perimeter. It's taking an average, it's creating a distribution of these pixel intensities. And what we're selecting is actually the middle 2/3 of our distribution and calling that our background. And the reason for that is we obviously don't want to subtract out signal from our background, but we also don't want to subtract out areas of the image that are lacking tissue. If there are rips in the tissue, or if your tissue doesn't completely cover your image, these might be spots that you want to eliminate. This isn't hard and set, though. What you can do is you can move these ROIs. So if for some reason, you want to make sure that these ROIs are quantifying the background a different way, that's great. You can also totally delete them out and just draw your own. I think typically drawing 3 ROIs from around your image is, pretty standard. So you can do, whatever fits your needs best. But there are a couple of different things you can do. To save this, I'm just going to press save. That goes back to my analysis window, and I'm set to go. So pretty straightforward. The last step here for my quantification is I'm going to approve my detections, approve my ROIs, and then I can export them. That's going to save in my image folder both a comma-separated value that it contains the quantification that I just did. And you can see I've done quite a bit of quantification in this folder. And it's also going to save a zip file that actually contains the location and boundaries of all the ROIs that I've detected in my image. And so let me show you those two things separately here. First off, let's take a look at our CSV file. I'm on a Mac, so I think it's going to open that. It would be nice if it opened it. There we go. So what I can see here in my CSV file are a whole lot of different data types. I've got, essentially a column for each one of the different quantification components, and I've got a row for each one of the cells that was detected in my image. And so from top to bottom, they're numbered 1 through 25, but I've also got an ROI ID number here in column C. And so that's pretty cool. The ROI ID, I think we should rename that ROID. The ROI ID there is actually going to be associated with an actual ROI ID that you can find in Pipsqueak. So over here on the very far right side of my analysis settings, I have an ROI table that has an ID number for each one of my ROIs. And this is great because I can click on it and actually see, these rows light up. And that's a really nice way to kind of audit maybe what I've done. I can go back and see the ROIs that I've collected. Or if you have someone else in your lab who's collaborating on this project with you, it's a really good way to be able to take a look at maybe what they have done or these cells that they've detected here, before going into stats. So, that's really nice. Outside of that, I've got a whole bunch of different columns here. Essentially, what we've done in Pipsqueak is made it possible to export any of the quantifications that you would typically take from your set measurements window in ImageJ. We wanted to make this something that's pretty familiar. So what we can see in here is, a lot of the different types of things that you would export, like your min-max area, integrated density, some things about, the shape of the ROI, like circularity, aspect ratio, roundness. So really anything that you're looking for. And these are all settings that you can set here in your project. So if I'm back in my project on the left side, I can right click on a folder and I can say export options here, or I can go into my image there and export options. So, pretty nice to be able to just tailor it to show you exactly what you want to see. Great. So yeah, we've taken a look at pretty much everything here that would be a standard single label analysis. The one last thing that I mentioned that I'll just show real fast is I've got a button down here for import ROIs. So what I'm going to do is I'm going to delete these ROIs and I'm going to press that import. And I'm going to go back to the file that we're going to go into a file. So this will be kind of fun. We'll see what we can find here. And I'm going to take some ROIs that I've apparently exported from another image. And so I'm going to import them. And what we're going to see here is that those ROIs pop up. This is, again, a really nice way to be able to audit what somebody else has done. But one of the really nice functions or features of this is it's the exact same format as you might be familiar with in ImageJ. So if you're performing your analysis in Pipsqueak and you would like to take your images and your ROIs over into ImageJ, take those zip files, take your image file, you can open them right up in ImageJ and it's going to look just like you did it in there. So, that's pretty cool to be able to go back and forth. Any questions that come up with that, again, feel free to reach out to us, or this is a good chance to pop in any questions about single label analysis here before I move on to the next thing that I want to show you. So pretty straightforward. It's really nice to be able to do a lot of this, in very few clicks. The whole idea with Pipsqueak is that we want to take something that, honestly used to take me like weeks to do as a postdoc and make it something that you could probably perform in just a couple clicks. Because, at least in terms of the workflows that I've performed, this is the last thing that I'm doing before, going on to stats. And I really don't want it to slow down my publication. So trying to make quantification as easy and painless as possible. So cool. Yeah, that's a single label analysis there. One of the things that I think is still kind of painful about single label analysis is when I want to do a lot of it. And so I've got an example here of, an experiment that I've done where I've collected, just a whole lot of images that are all actually the exact same image type. So let me go to one that I haven't analyzed. So, what you can see here is, again, a lot of that WFA staining. And there's, probably, I don't know, 50 or 100 cells in this image across a whole lot of images. So rather than have to go through and do each one of these images one by one, we've created some batch functionality that will hopefully speed that up for you. And so it's pretty cool. You can do this in a number of different ways. It's really using the exact same single label workflow that I just went through, except I can choose multiple images at once. And so I can either select an image, hold Shift or Control, and select the images that I want to do that way. I can go up to the folder that all these images are in, and I can just right-click there and say batch detection. One way or another, I can open up all these images all at once. And so what you can see now is I still have some of the same analysis settings that we've seen in single label analysis. So over here, I can again choose one of my detection models. So I'm going to choose WFA because I remember that that's the one that works well for these images. I can choose my sensitivity settings, my overlap setting, and press batch. And so what's going to happen here is while we're talking, it's going to start going through and analyzing each one of these images just kind of sequentially. On your left panel here in your project settings, you'll see each one of these images process. And as it finishes up, you get a check mark. So pretty easy to see this happen. And this is really nice if you've got, a whole lot of images. We actually just ran an experiment the other day where I think we had like 1,000 images in this thing. And so, I set it up, I pressed detect and went and got a copy. And by the time I came back, all of my detections had been completed. So that's really cool. This also still allows me to go in and make that certification of the detections that it's captured, because, as a scientist, I'm not going to take its detections blindly. I want to make sure that I agree with what it's detected here. And so for me to be able to do that, I can click into any one of these images. I can, again, draw on it, draw the cells that I would want or change the cells. I could go back and I can reanalyze this image in particular if I wanted to do that. And I can basically do that for everything that I've batch processed. Once I'm finished with that, all I have to do is, again, press approve ROIs. You'll see these check marks now turn into a check mark in a square over here on the left, which basically means that the system has created that ROI file for these images, and I'm ready to batch export. And so I can either export my ROIs there, or I can go over, I could even right click on one of my folders again, right click on a folder here and press, export ROIs. And so this is kind of nice because what this export has done, I think I clicked it wrong. Yeah, what this export is doing is it's creating a single comma separated value file, CSV file, with all of those data inside of it. So this is really nice if you want to, process your entire experiment and then go back and start to, clean these data up, get them ready for stats, all in the same file. So what I'm seeing now is, in addition to all the columns that we've seen previously, I also have a column here for my image index. And that's essentially telling me that I've got, this 70 cells detected in image 1, and then another 50 cells detected in image 2, and so on all the way down. So again, really nice to be able to start putting your data into the shape that you need for stats. If you want to collect subject means or image means, this is a great place or a great way to start to get to that point. So I didn't see any questions on single label analysis. Any questions on batch processing or anything that I've shown so far? I assume I'm just doing an incredible job of explaining it, so I understand why there are no questions. But great, yeah. So this is, 90% of the functionality that I need to be doing in Pipsqueak is really covered by all these things that I've shown you here. One additional feature that I think is super useful is the ability to start filtering the ROIs that I've collected. And so I'm going to go to a slightly different stain type here. This is a stain for parvalbumin, calcium binding protein. So again, I'm going to going to detect these cells. I'm using the parvalbumin model here. This again would be a good time to choose recommend a model if I didn't know how to do this, but great. I kind of like these detections. And so I've detected these, but let's say I don't like having oval or rectangle detections around my cells. I want something that's actually segmenting my cells. Well, we've got a couple options for you. The first here is over on the right side. I've got down at the very bottom, this ROI filters tab. And in my filters tab, I have a couple cool things. One, I can filter ROIs. And so this is pretty cool if I wanted to, filter out the detections by maybe the area. I could say, well, my minimum area, I know these cells aren't smaller than 50 pixels across or something. And so I can start, increasing my area until I've filtered out everything that's smaller than that. That's pretty cool. So take a look at this region right here. You'll see an ROI pop back up as I take the filter off. Yep, so that kind of gives an idea how that works. I'm going to clear those filters out, and I'm going to show you how to segment these. This is a really neat feature that allows you to segment either or based on intensity, either from the top or from the bottom. Here, I'm going to go from the bottom, and basically I'm just going to bring this minimum intensity threshold up until I've kind of got a nice contour around the cell that I'm looking for. So, this ended up working pretty well for some of these. I kind of like what it's showing there. Oh yeah, that's looking great. So I can apply that modification and, now go back and finish up my my quantification if that's what I'm looking for. This is a nice tool for segmenting images. Something that we have coming down the pipeline that I'm really excited to share is the ability to use different types of AI detection models that are purpose-built for segmentation of images. And so, you've used things like, Cell Profiler or maybe some of the Nikon NIS tools. This is maybe more familiar in terms of how your objects are being detected. Segmentation works great in a lot of scenarios, like especially this scenario here where we're seeing, really round, kind of standard looking cells. Segmentation doesn't work as well in scenarios where you have hard to define cells. I think microglia doesn't segment very well. These WFA or this WFA staining certainly doesn't segment very well. So, it's really just kind of finding the right tool for the right scenario, and we're going to make it possible for you to choose both. So that's, I think, a really powerful thing that you'll see here fairly soon. So yeah, Ahad, let me, oh, great. I think Christy answered your question there.
I beat you there, John.
yeah, thank you. Let me know if you have anything else that you want to know about that. Cool. So yeah, that's a really neat functionality. The other thing I want to show off here is the ability to do co-localization. So what I've shown you so far was the ability to analyze a single channel, but in reality, all of the staining or all of the analysis that I was doing is from a confocal where I've stained tissue with multiple markers simultaneously. And so in this case, this tissue was stained for parvalbumin. It was stained for WFA. This is an extracellular matrix staining protein. And then I think in this case, it was CFOS. And so in my co-localization workflow, I can basically choose the right detection model that I want to use for each one of these. I can then open up a merged image if I have it. And this is a nice way to visualize the detections that we're going to get. Or let's say I don't have a merged image. And so let me show you what that looks like. Down here, I'm going to select just the 3 channels I want to use, or maybe even just 2 channels that I want to use. We'll use all 3. I can right click on that co-localize. And now, if I don't have a merged image, I can actually create one. Just press this green button up there and voila, it's actually built a false colored image from the images that you've shown it. I think it's pretty cool because this merged image is actually, I think, a little bit cleaner than the one that we were exporting off of our Leica microscope. So, use what you want to use, but this is a nice way to produce it if you don't have it. So, all right, let's continue with this analysis. I'm going to, again, just put the detection models that I want to apply to these. And all I have to do is press detect cells. So similar to that batch processing module, this is going to detect each one of these cells or these channels simultaneously. So great, we've got each one of those. if I wanted to go in and change anything, I can click into each one of the images, draw my additional ROIs or whatever I'd want to do, just like I've shown you. And then finally, I can co-localize here by running my co-localization function. And that's basically going to show each one of these, in this case, triple label co-localizations on my merged image. There are a couple of different types of co-localization that we make possible here. So if you're looking for multiple objects inside of one object, that's possible. If you're looking for adjacency, that's possible. It's also possible to look at just maybe like a double label co-localization here. And one of the best ways to do that is simply to press skip a channel in this process, and that's going to focus in on the other two channels that aren't skipped. So just like with my other analyses, to finish this up, all I need to do is press approve, and then I can press export, and that's going to give me, again, another one of these CSV files. This one looks very similar to everything that we've been looking at so far, and similar to my batch processing, this one shows me all those different images here in a single file. And so that's kind of nice because I can see my PARV, my WFA, my CFOS all together. And what it's showing me here are subpopulations of these cells, either that co-localize. So I'm sorry, you're seeing that because they are co-localized with the other two channels, or cells that are not co-localized. And those are indicated because they have no co-localization present. So again, going back to auditing, this is pretty neat because now I know that PARV cell number 9 actually overlaps with CFOS cell number 13 and WFA cell number 10. And so if I go back here, I can open up my ROI table and I can start to find maybe exactly that one cell that's, I don't know, an outlier in my data set or something that seems weird. So you really have that ability to dive in and dig into your data, to understand what's going on there. So pretty cool. Again, all the types of quantifications that you would want. We have a fair amount of documentation on our website and in our tutorial here on what each one of these metrics are telling you. If you have questions about it, again, please, just email, reach out to us. Happy to dive into it and, show you, how that quantification is being produced or anything like that. So yeah, pretty powerful there. So before I move on to the last thing I want to cover, just any questions on co-localization, on batch processing, on single label analysis. Hopefully this is looking pretty familiar to you. Again, these are tools that we've kind of based off of maybe some of the workflows that you've done in like ImageJ. So, we want this to be fairly intuitive and pretty straightforward. So, yeah.
Can you indicate if the co-localization, up to what degree of co-localization? How do you define the edges of co-localization or the boundaries of co-localization?
Yeah, that's a great question. So our co-localization here is based off of using our this overlap slider. So essentially what we've developed is in this type of object-based co-localization, it's just saying how much overlap is necessary of these pixels for your co-localization to be considered co-localization. So I can see an example down here where this was a rejected co-localization because these cells are, I think, not overlapping quite well enough with the WFA. Similar here, these are falling into the parameters. So you can adjust this a little bit. We do have some documentation on this that I'd be happy to send to you. Again, these are based off of the types of co-localizations that you might be doing in ImageJ.
A second question that I have for you is also the relative intensity of the different markers. So are those defined independently for each channel? So what you define, how do you cut the pixels so that you define intensities, because that can change depending on where you put your background for each marker.
Exactly. Yeah, that's a really good question. So our background subtraction method is, really kind of taking some of that into consideration. So, the background subtraction that I might be doing out of this PARV image could certainly be different from the WFA and the CFOS image. So what we're reporting back to you is, that arbitrary unit as just a pure number, but we're also providing a mean minus background column in this that is, a normalization that we would always report in some of my work. I think, either using that or an integrated density metric are, two good ways to kind of get around some of the variations in intensity or just variations in staining across your experiment. Certainly applying the same background subtraction grid and rolling ball diameter to your images, I think is a good way to make sure that that's standardized. But those are some parameters that I think vary a little bit between labs, and you'd certainly want to make sure that you're doing that the way that you feel comfortable with.
And nobody else has got another question. Can I ask another question?
Feel free, yeah.
Then also with regard to what is your recommendation with regard to the Z thickness? Because that also makes a difference to defining localization.
Great question. Yeah, thanks for reminding me. I actually kind of glossed over this and forgot to mention it. So all of the images that I've shown you so far are collected from a confocal. Pipsqueak works great with fluorescence microscopy or even bright field, so any of that's fine. A lot of the work that that I did in my postdoc looked at Z thicknesses of around 10 microns. We've certainly worked with labs that are much thicker or much thinner than that. We don't have any problem with that. I think what you start to see is overlap of cells at some point, but the one thing that I do recommend to our users is to make sure that you're summing your images rather than taking a max projection of that Z stack. And the reason for that is a max projection is essentially pulling out just the brightest pixels from each one of your Z steps and showing that in your final image, where summing it is going to create a relative pixel value across all of your images. So if you're looking to quantify intensity, I think summing is the way to go. We've got some functionality for you to do that in here. What you can do is essentially in your projects, go to your import functions. You can import a stack. And so this is pretty neat. We've again designed this to look very similar to ImageJ in that it's, let me see if I can find this here. So yeah, I can choose the image that I want to import. This is a Z project that has 4 channels in it. So I can indicate that by saying the number of steps. So that'd be 4 in this case. Oops, not 14. And I can also choose which index I'm going to start on. So if I want to import the merged image that you already saw there, I'd start with 0. If I want to import the next channel, I'd start with 1. And so I can import this, and basically what you'll see here is great. This looks like it was the parv channel. So you can kind of start to import all these different projects that way. Currently, if I analyze this image, I can do that by applying my model, press detect. That's going to analyze the summed image. And I can, oops, if that pops back, there we go. I can have that show me those detections here on the Z plane, which is kind of nice because as we talked about, you start to get some of that cellular overlap. This is a good way to maybe actually see where that happens. Another functionality we have coming out here very soon is the ability to actually analyze each one of these Z steps independently. And so what it's going to do for you is start to develop a 3-dimensional detection of that object. So you'll see your detections start and stop within your Z plane. And hopefully that provides maybe another level of analysis that you can't currently get here. So yeah, that was a great question. Thanks for asking that. But I guess I didn't give you a recommendation on size. The program is capable of looking at anything you show it. So that's just the limitation. It would really just be more the experimental question you're asking. So yeah, pretty cool there. Where I want to end today, unless anyone else has any questions coming up here, is just what to do if you've got an image here that maybe doesn't work very well with our detection models. So maybe you have a different stain type, you've got a different protocol than what these models have been trained for. So this is going to be a scenario. Let's go back to one of the batch examples that we were looking at here. Oops, there we go. Great. So here's one of those WFA images that we looked at. Well, if I, if this was just a poorly fit model and I ran a detection, what I might see is a whole bunch of false detections come back. And this would be a scenario here, for example, that I'm just not very happy with. So what Pipsqueak is really designed to do is enable you to tailor the detections to be accurate, to edit the detections to be accurate, and then train the model to reproduce that next time around. So the way that I can do that is I might start with, a set of detections like this. Let's say this got me, close to what I'm looking for. Well, I can go in and I can clean this up a little bit. So here's one tool that's really handy. I've got a select function. So I'm going to maybe select all these ROIs that I guess I don't like, and I'm going to delete them out. Ahad, feel free to hop in here if you have a question on this.
Yeah, I had a
Yeah.
quick question on like custom model building. I know it's slightly tangential to what we're talking about right now.
Yeah.
But
No, I think
is there a way to refine existing models instead of starting over every time?
Yeah. Great question, because I know you guys were working on that a little bit. Let me answer that when I get over to just the next stage of this here, because I think that's a great question.
Sounds good. Thank you.
Yeah. So, essentially what we're doing here is we're developing a training set of images, that I'm going to apply to my model. So I would go through, I'd make sure that I'm detecting exactly all the cells that I want. The trick to this is that I need to make sure that all of the detections I show our AI trainer are exactly what I'm looking for, because, really kind of the mantra in AI is garbage in, garbage out. If you, are showing this bad examples of what you're trying to find, it's going to reproduce that and probably give you, sub-adequate results back. So I really want to make sure that these are great. So in this case, let's say this is what I've come up with. These are great. I really like it. I've approved my ROIs. Well, now to start training a custom model, all I need to do is go over to the right side here, click this second icon down, custom models, and this allows me to create a model. I can name this, whatever I want to name it. What's today, the 19th, and give it a little description. And then I just need to associate this model with one of the projects that I'm working on. And the reason I do that is because the the project I'm associating with it is where we're going to grab the training data from. I can also choose to share this model with anyone that I'd want to share it with. So, I might want to share it with Christy. And just by putting in her, however you spell Rewire, that's fine. Putting in her email there. And that would add her to this model so that when it's trained up, she's going to see it pop up in her library as well, which is pretty cool. That's not Christy's email, so I'm going to just delete that. So great, I'm going to create this. When this pops up over here, I now see it down on the bottom. I can click into it, and all I have to do is add images that I've annotated perfectly to this. So again, hopefully this is something that's just a byproduct of your analysis. If you've gone through and analyzed a minimum of 5 images, like we've been looking at, that is technically sufficient in order to start training an image. So in this case, here's an image that I didn't do very well on. So that's actually a bad one. So I'm going to deselect that image. I don't want to add this to my data set. But then I'll, that one's actually a different data type, right? I don't want to include that. But, at this point, maybe I've got a lot of good examples otherwise that are things that I want to add to my image or to my model. So I'm going to press add. That's putting these, probably 20 some images into my training set. I've got 30 out of the 5. So this is great. And this is really kind of going to the point of what we're looking for in model training. We need a minimum of 5 images, but the more examples you show the AI trainer, the better the AI is going to do. Really, these type of deep learning AI models need thousands and thousands of examples to look at. We've created some really fancy tools that essentially are, taking your small data set and learning from that because of some of the stuff it already knows how to do. So, this is a nice way to make it easier for that training. So I've taken those, at least 30 there. And now what I need to do is just draw a contour around some of these cells that I'm looking for. And this is a good example or a good chance to not only see how well my annotations are placed. Like in this case, I don't think this is a very good annotation, so I'm going to actually delete that one out. But I can also, I can also really kind of get a feel for the data that I'm putting into this. And so all I need to do here is draw the the best contour I possibly can around these objects, press approve, and then move on to the next one. And so what we'll see is my little contour counter is going to start counting up here. I'm going to skip this one because I don't think that's a good example. Actually, that's a kind of neat example here. Rather than skip this one, I'm just going to adjust my ROI a little bit. Great. Now that fits it better, and I can draw that good contour around it. Obviously, you'll do a better job drawing contours than I am because I'm going fast here, but this is essentially the idea for that. So cool, we've gotten all the way up to at least 20. If you want to do 100, that's wonderful. The more you do, the better. And when you get to that point of at least 20, this train model button is gonna pop up. And let's see, I think I've got an example of one that I can show you here. No, not that one. Anyway, so yeah, our train model button is going to pop up, and that's going to essentially give me a warning here that I'm about to start training this custom model. Typically, this takes around a day right now to run. What's happening is your images are being pulled onto our machine learning server. It's running this big ML experiment and training up a bunch of different options and deciding which is best for this scenario. So that typically takes about 24 hours. Again, we're making a lot of changes to the system. So if you run into a delay with it, feel free to, just email us, ask what's going on with it. But it's a fairly automated process. So when it's done training, you're going to get an email that says, Christy, your custom model is now available for you in your library. And so if you scroll down, what you can see is that that custom model now present in your library. So going back to Ahad's question here, what do you do if the model that you trained really isn't what you're looking for, if it didn't perform as well as you'd like it to? Well, there's a couple different things that you can do. The first thing you can do is just keep adding images or go back and refine the images that you've included so far. So if I'm in that project that we just started today, this might be a chance for me to add more images, draw some more contours, or maybe go in and look at the images that I've included here and remove some that aren't good examples. If I press train again, what's going to happen is it's actually going to start over, and it's going to retrain this model from the ground up. It's going to deploy it, and it's actually going to write over the last one that we saw. So if you would like to hold on to versions of your model, this is not currently the way to do that. Another thing that I can do is I could go back and I could start a second model. So if I'm in my custom models tab, I can just press create model again. I can choose that same project that I've been working on here, and I can start a new one with a different name. And this is a really nice way to start to version your models. So if I add all the same data plus some new data, then this is going to be, maybe a little bit different, maybe a little bit better than the last version. And so that would be a way to kind of compare them. Something that we're working on here in the hopefully near future is the ability for you to start tuning your models, more specifically from some of these save points that you've created. So if you train a model and you'd like to go back and tweak it, one thing we're working on is for you to be able to say, I want to just retrain this model, maybe try it again with some different parameters. So not something that you can do right now, but Ahad, hopefully that gives you an idea of what we're working towards, where this is going. So yeah, great question. So I have been droning on for a very long time. Hopefully this has been helpful to see a lot of the different things that this program is capable of doing. Really our thesis is that we want to make Pipsqueak as powerful and as broadly applicable as possible. This is really designed to perform detection and quantification capabilities across a lot of different image types, across a lot of different staining types. So, hopefully this is something that you think would be useful to you and your workflow. And again, if you have questions that come up after this, or if you would like to walk through some of these, these steps or these workflows with your images, feel free to reach out to us. I'd be more than happy to set up a one-on-one demo with you or with your lab. We can go through some of your images and look at how Pipsqueak performs. There are a couple links here you can hit with your camera if you'd like to scan the QR code or just go to this URL. And we are going to make this recording available after today, so you can go back and find this later. But to get started with Pipsqueak, I already showed you here on our rewireneuro.com. Or if you'd like to set up that demo, here is our HubSpot link where you can find a time that works for you there. So yeah, this is a really powerful tool. We like to talk about it. Christy writes a blog every now and then that you can find here on our website. So, check that out. We've got a couple good articles here that actually talk a little bit more about some of the machine learning behind the scenes and how we're training these models. So if you're curious about how that works, certainly AI is in vogue right now. So you can go see how we do AI versus ChatGPT, I guess. But you can find that here. I'm happy to answer any other questions that come up today, or feel free to reach out to us after the fact.